Alrighty. So today we are continuing on with the nervous system, and we are going to be looking at the brain and the different parts of the brain and what they do, and then we'll look at cranial nerves. Cranial nerves is something that you will also see in lab. The first time you see cranial nerves, it seems like kind of a lot of information because there's 12 of them, but pretty much everything that we're going to look at today is, again, not a terribly complicated physiological process. We're kind of just looking at a thing and naming what that thing does. So doesn't mean it's easy, but it's straightforward at least. So it shouldn't feel complicated. It's probably just a lot of new information. So before we get into the parts of the brain and what they do, first we're going to talk a little bit about how these neurons work. So today's Thursday. On Tuesday, we talked about neurons. We met the neuron. We named the parts of the neuron. The neuron is just a fancy name for a nerve cell. The job of the neuron is to generate an action potential. So the neuron is a nerve cell, resides in the nervous system. The neuron is able to pass these electrical signals along to the body. If we're looking at muscle contraction, the neuron is the cell that sends that action potential that tells the muscle to contract. Here we're going to look at how is it that that nerve cell works. We're going to go into a little bit of detail here, nothing like the detail we saw in the muscles though, but it will sound maybe a little bit familiar. So a neuron, a nerve cell, when it is at rest, so when that nerve is not actively firing and sending a signal, the internal charge of that neuron is really, really negative. If we are measuring the charge in millivolts, the resting charge is negative 70 millivolts. And that's the resting state, and we would call this polarized. So when a neuron is at rest and it sits at its super negative state, it is polarized. When the neuron is going to send its action potential, which will be a message to some other cell, it goes through the process of depolarization. That word should hopefully sound familiar. Depolarization is the same process that occurs when that neuron sends the action potential to the muscle cell. And it means the same thing. Depolarization is when the inside of that neuron suddenly becomes a lot less negative. You could also say that it becomes positive. So the process of depolarization, or when the neuron becomes depolarized, is when the internal charge of the neuron changes from its resting state at negative 70 to suddenly becoming much more positive. That neuron will pass a threshold. The threshold is at negative 55 millivolts. What that means is, once that neuron starts to become less negative, so we're going from negative 70 to negative 55, so it's less negative. Once that neuron becomes less negative and we reach negative 55, that's our threshold. At that point, that neuron is definitely going to depolarize, which is it becomes rapidly more and more positive. We get up to positive 40 millivolts. This is significant because that depolarization is what allows the neuron to send the action potential. So the depolarization is what allows the neuron to send the action potential. Once we pass that threshold, it's definitely going to happen. That is called the all or nothing principle. The all or nothing principle is it doesn't matter what really happens after we reach this threshold. As soon as we hit negative 55, it's definitely going to go. It's like when, this is a classic example, is when you squeeze a trigger on a gun, which actually I haven't said that out loud in a while. I really don't like that example, but I can't think of a better example right now. I will think of a better example at some point. But the classic example is when you squeeze a trigger on a gun, and the reasoning is it doesn't matter how much force you place on the trigger. Once you get just enough force to move the trigger back, that gun is going to shoot. It doesn't matter if you're gripping with superhuman strength or if it was for some reason really hard to pull that trigger. You just need enough force to overcome that trigger resistance, and then the gun will shoot. Does that analogy make sense? That's the all or nothing principle. These next two slides are just visuals of what happens during that process of depolarization. This is very similar to what happens when we were looking at that signal being sent from the motor neuron into the muscle cell. So over here, this picture here, this is when our neuron is at rest and we are polarized. The inside of the cell is very negative 70 millivolts. And at this state, our sodium and our potassium gates are closed. Okay, that's our resting state. When depolarization starts to happen, this is because the sodium gate opens and sodium enters the cell. This hopefully also sounds a little bit familiar. Why does this change the internal charge of the cell? Guesses? Yes?
It's because the sodium is positive.
Yeah, awesome. The sodium has a positive charge. So because sodium has a positive charge, getting a whole bunch of sodium inside the cell is going to become more positive. That is what depolarization is. And here we go, we can see we're starting to become more positive. Once the inside of that cell reaches negative 55 millivolts, we've hit our threshold, and the cell is going to rapidly depolarize. At that same time, the potassium gates open. Okay. Then, after depolarization has happened and repolarization begins, repolarization is when the cell starts to go back down to its resting state. Repolarization begins when the sodium gate closes and sodium is no longer coming into the cell. But our potassium gate is still open, and now we have potassium leaving the cell. So we have positively charged ions leaving the cell. That is going to make the cell less positive. You could also think of it as more negative. And that will return us down to our resting state. That is called repolarization. So depolarization is when the cell becomes positive and we send the action potential. Depolarization is when the cell becomes positive and we send the action potential. Repolarization is when the cell returns to its negative resting state and we have a little rest period. We're not sending an action potential. Yes?
It looks like that repolarization is more negative than its resting state.
We have a refractory period, which is what happens after that action potential is sent. And we definitely talk about refractory periods at some point. I can't remember where exactly I have that on here. I think I might have it on the next slide, actually. Yes, this is the same thing we just talked about, just a different visual, in case this one clicks better in your brain. Okay. So at our resting state, the neuron is very, very negative, negative 70 millivolts. Depolarization starts with the sodium channels opening. Sodium, with its very positive charge, comes into the cell, starts turning the inside of the cell positive. Once we hit that threshold, we pull the trigger, so to speak, and we rapidly depolarize, which allows the neuron to send the action potential. So sodium channels open, sodium comes in, potassium channels open. Then, after that depolarization, the sodium channels close. We don't have sodium coming in anymore, but the potassium channels remain open. So we have potassium leaving, which also has a positive charge. Because potassium has a positive charge and it's leaving, we are losing positivity, so we're becoming more negative. And then yes, as Harper pointed out, after that depolarization and the repolarization, we have what's called a refractory period, which is where we dip a little bit below the regular resting state. And at this point, this is like the rest period where we need to take a break, and we can't start working until we finish that refractory period and go back up to the regular resting state. So knowing the terms depolarization, repolarization, what they mean, knowing the actual number of what the resting state for neurons are, and which ions are involved are the types of things you'll see on the exam from this. Okay. So we have successfully depolarized a neuron. The neuron has shot off its action potential. Now what happens? That action potential travels down the axon. We looked at axons on Tuesday. This is just the name of the little highway on the nerve cell. Here's a picture of a neuron. Here we can see the axon. In this picture, the axon is wrapped with little hot dog buns. Those little hot dog buns are called myelin sheath. We'll look at this in lab also. The myelin sheath is like a wrapping. When you're looking at electrical cords, that wrapping around the electrical cords is like the same idea. It protects it, it coats it. It also allows for the signal to be sent much faster. So we have different types of neurons in the body that are either myelinated and have the little protective hot dogs, or unmyelinated. When signals are sent across an unmyelinated axon, they travel slower. So this would be continuous propagation. The signal is still sent, it's just sent in like little small steps. Compared to when that action potential is sent across a myelinated axon, it goes super duper fast. This is called saltatory propagation. And you can see the difference, 2 miles an hour for continuous propagation versus 40 to 300 miles an hour, so really quite a bit faster. The difference is the myelin sheath, and it allows for big leaps to happen from node to internode. So a node is like the end of one hot dog bun. You see how it kind of pinches off there a little bit. The message can basically jump across, so it goes much faster. Interesting side note, there are diseases that attack the myelin sheath that really disrupt the nervous system and voluntary motor control. Does anyone, this is random, but does anyone know like what one of those diseases would be? I didn't expect anyone to. Do you have a guess? Do you want to guess? No. Do you want to guess?
Alzheimer's.
Alzheimer's. Oops. Yes, Alzheimer's, and also multiple sclerosis. You guys familiar with multiple sclerosis? I used to work with MS patients when I started off doing group exercise instruction, personal training at the Monterey Sports Center, and I taught a group exercise class for people with MS. And MS affects people on a really wide range, but the myelin sheaths are attacked. And so this can present, again, huge range where some people have pretty good control, and every now and then they'll have a bad day and walking is kind of hard, whereas other people are completely wheelchair bound. So it really depends person to person and also how severe it is. But where movements for non-affected people would be fluid, things like buttoning a shirt, putting on shoes, brushing teeth would be really hard because this protective layer around the axon is being attacked. So that signal that's being sent gets kind of clunky and mixed up along the way. Okay, when neurons communicate to other neurons or to other parts of the body, they use a neurotransmitter to send that signal. We know a neurotransmitter already. Does anyone remember what neurotransmitter is used when a motor neuron tells a muscle fiber to contract? Yes, ACH. And what does that stand for?
Acetylcholine.
Nice, acetylcholine. And that should be up there. Here we go, ACH. ACH is a neurotransmitter. Other neurotransmitters, norepinephrine, epinephrine, dopamine, serotonin. These words might sound familiar. You might recognize these as hormones. These are in fact the same chemicals. But when the nervous system uses them, we call them neurotransmitters. When the endocrine system uses them, we call them hormones. And we'll take a look at hormones at the end of the semester. So, when a neuron wants to send a signal to any other cell, it has to use a neurotransmitter. That neurotransmitter is sent out to the neuroeffector junction. Neuroeffector junction is just a blanket statement for the neuron meets something else. When the neuron meets a muscle, it's called the neuromuscular junction. So neuroeffector junction is basically just the end of the neuron where the neurotransmitter will be sent out. That signal that the neuron sends is either going to excite the next cell, meaning pass that message along, or it is going to inhibit the next cell and stop something from happening. That's a very basic general statement. The way that signal is received is dependent upon the receptor cell. And we will talk about specific examples as they pop up. But the example that we're familiar with skeletal muscle, that would excite the next cell, meaning make that muscle contract. Excitatory. Inhibitory is making something stop. So neurons can communicate with other neurons. They can communicate with other types of cells. And we have more efficient ways than just one neuron talking to one neuron and talking to one neuron and passing that message along. We're able to combine groups of neurons into what's called neuronal pools. We have lots of interconnections. And there's two different categories. Divergence would be one neuron is going to spread the information out to several other neurons or several other cells. And the opposite of that would be convergence, which would be several neurons that are receiving the information converge onto one neuron. That would be like receiving sensory information, converging onto one neuron, and then passing that to the central nervous system. Okay, now we are at the what are the parts of the brain and what do they do? So if you haven't looked at the brain ever before, this might all be new information. This whole section make really good flashcards. This is kind of a straight memorization section. We're just going to go through it and talk about what each piece does. So the brain in general, when people think of the brain, you're probably thinking of something like this picture here with the squiggly, maybe thinking of it as pink. That squiggly pink part is the cerebrum, but there are other pieces of the brain as well that we'll look at today. The diencephalon, the midbrain, the pons, the medulla oblongata, and the cerebellum. We will also look at this in lab. You're going to become very familiar with these different pieces. So don't worry too much if right now it seems like so much information, because you will get a chance to look at something like this enough to where you're able to see what the separate pieces are. Okay, so the cerebrum. This is the largest part of the brain. This is the pink squiggly stuff, not the stuff here, but just the pink squiggly stuff. And the cerebrum is separated straight down the center by a dividing line called the longitudinal fissure. It's just the name of the line down the center. And that separates the cerebrum into two sides that we call hemispheres, right and left. That's simple, just right and left hemisphere. The two hemispheres are able to send information back and forth to each other using a structure called the corpus callosum, which we can see is this thing that kind of looks like an ear. The corpus callosum here. That connects the two sides. The wrinkles, all of the squigglies that we have in the cerebrum are extremely important. Those squiggles greatly increase the surface area. So instead of it being smooth, all those inward foldings allow us to basically pack way more brain into our skull. So we have a lot of brains, lots of neurons, lots of surface area. Question or comment?
Yeah, just fun facts.
Yeah.
All those do not have folds in their brains. They are smooth, so they are really stupid.
Koalas. That's poor little koalas. Well, they are very cute, though. I guess there's that at least.
They're also susceptible to chlamydia.
That's very interesting, and I did not know that either. Thank you. Because we have to name everything in anatomy, we name the squiggles as well. So the squiggles are made up of gyri and sulci. Gyri and sulci, that's a plural. The singular would be gyrus and sulcus. You'll see these terms on your terms list. These are referring to specifically the indentations, the indented portions are the gyri. The sulci. Remember, a sulcus is a groove. Remember that from the skeletal system? And then the parts that are raised up are the gyri, or gyrus if we're looking at one specific raised portion. And then within the hemispheres are separated into lobes. And this should sound refreshingly familiar. The frontal, temporal, parietal, and occipital lobes. They're named for the bones, or the bones are named for the brain. Okay, so we're going to talk about each lobe and what it does. The frontal lobe here is the primary motor cortex. So this is where we are controlling somatic motor sensations, so skeletal muscle, stuff that we have control over. Also in the frontal lobe here, we have our gustatory cortex. Anyone know what sense gustatory might be referring to? The sense of gustation? Taste. Okay, so frontal lobe, motor cortex, and taste. Parietal lobe, right underneath the parietal bone, sensory cortex. So all the different pieces of sensory information, and we'll look at sensory information next week closer. All the little pieces that we feel, tickle, temperature, vibrations, all of that information gets sent to the sensory cortex here. Occipital lobe in the back, this is our visual cortex. Everything that we see lands in the occipital lobe. And then temporal lobe, this is where our auditory and olfactory information lands. Auditory is hearing, olfactory is smell. Okay, so again, I would suggest flashcards for that. There's definitely going to be questions on the exam about which lobe receives which kind of information. Okay, now, those different lobes are basically the landing point for that type of information. So visual information is connected straight to the occipital lobe. Sensory information straight to the parietal lobe. It just lands there. But then we have association areas. Association areas receive information from the primary motor cortex, the primary sensory cortex. It receives information from all of the original landing spots, and it puts them together in a way that we can understand. So when we are looking at a cow, we see the cow with our eyes. That visual information lands in the occipital lobe in the visual cortex. Maybe we can hear the cow. Maybe it's mooing. That lands in a different area. Maybe we can smell the cow. That goes to a different spot too. Maybe we're touching the cow. So all that different bit of information, it lands in its respective spot, and then that gets sent to the association area, and then we go, aha, it's a cow. And then all the information is put together, and we know and we understand fully that it's a cow because we can see it and touch it and hear it and smell it, and you could lick the cow if you wanted to taste it, and then you would be receiving all sorts of sensory information about the cow. Okay. But that is still fairly basic information. The next step up from the association area is called an integrative center. The integrative center gives us a higher understanding of what's happening. So information from the association areas gets sent to the integrative centers, and then we are able to communicate about the cow, or read about the cow and write an essay about the cow, or understand that if we don't feed the cow is going to die. More complex things than just saying it is a cow and I know that it's a cow. So information from the various association areas gets sent to integrative centers. We have different integrative centers that allow us to do slightly different things on, again, kind of a higher level. So Wernicke's area, this is our general interpretive area. Wernicke's area allows us to interpret the sensory information that we are receiving and be able to make sense of what we are hearing or reading. Damage to Wernicke's area presents with a very interesting type of what's called aphasia. Aphasia is when something affects the way you're able to talk. So somebody that has damage to Wernicke's area is not able to integrate that information. So nothing really makes sense fully because they're not integrating the information, they're not fully understanding it. Whereas Broca's area, this is our speech center. So this allows us to regulate our breathing in order to speak normal speech. Damage to Broca's area gives us what's called Broca's aphasia, which would be essentially somebody that is just physically having a hard time speaking. They understand what's going on. They completely understand what's going on. They just can't physically get the words out. Wernicke's area, that person does not understand what's going on, but they can get the words out. So it's, fluent aphasia is interesting because a person with damage to Wernicke's area does not understand what's going on, but they can speak totally normal. That would be like me up here saying, chair, fire, rainbow tree, troll, gummy bear, bed, hair, shower, landfill. And I know it's kind of funny. It's not funny, but it's my example is maybe fun. So I sound like I'm speaking totally normal. My pattern and rhythm and my breathing and all of that sounded normal, and I could go on for days and days because my speech center was fine, but my understanding was not there at all. That's damage to Wernicke's area. Again, damage to Broca's area is you can understand everything, but you can't actually vocalize it. Does that make sense, the difference between the two? So for me to be able to stand up here and talk normally in a way that actually makes sense, and then actually understand what those words means that both Wernicke's area makes me understand everything, and Broca's area makes me able to say everything correctly are working, the integrative centers. Questions on that piece? Does that make sense? And then we have the prefrontal cortex. This allows us for abstract thinking. This is what allows us to predict consequences, like, gosh, this is a whole bunch of information. If I don't study this, probably not going to do that well on the exam. Allows for predicting consequences. This is the area that's removed in a lobotomy. Really old movie called One Flew Over the Cuckoo's Nest. Has anyone heard of that? I don't think so. It's old for me too, so I don't feel too bad. More recent movie, but probably still a little old, Sucker Punch? No, nobody. Okay, well, movies that had people that had lobotomies. Which was a procedure that was performed years ago on certain people, and I should have read up on it a bit before lecture, but certain people with, I forget what the diagnosis would be for a lobotomy, but people that would be like declared insane to some degree, they would perform a lobotomy and then they would suddenly be like at peace, like, wow, everything's great, and like kind of like a vegetable. That's because they're destroying that prefrontal cortex. Okay, an electroencephalogram, or EEG, okay, kind of sounds like electromyogram, or EMG, which we did in lab last week. This is basically the same idea, but instead of looking at the electrical activity of muscle, we're looking at the electrical activity of brain. Right? And it's done the same way with electrodes placed on the surface of the skin. People will have EEGs done when you need to monitor brain activity. If you ever have a sleep study done, they'll hook you up to an EEG as well. You can actually see brain waves when you are sleeping, rather. When you're awake and conscious and actively paying attention in class, this is probably what all your brain waves look like right now. Lots of activity. When you're awake but calm and not thinking, we see the waves start to spread out a little bit. When you're in deep relaxation, the waves spread out even more, and then sleeping, the waves aren't as tall and they're spread out even more. Question?
Yeah, similar to the EMG, right? More force to the closer.
Yeah, absolutely. Yeah. So the amount of electrical activity is going to be the height of the wave, and the frequency is going to be how close together they are. Yeah, very good. Okay, continuing on with the brain, the diencephalon. This, if you were to cut the brain in half and then take one half and turn it on its side, that's what we would be looking at in this picture up here. This is called a mid-sagittal view. So again, cut straight down the middle, turn it to its side, and that's what we're looking at. So the diencephalon is this guy right here. This is an important area of the brain that integrates all sorts of information, conscious and unconscious information. We can break the diencephalon down into three smaller pieces: the epithalamus, the thalamus, and the hypothalamus. These are just areas within the diencephalon. So the epithalamus contains a special gland called the pineal gland. The pineal gland produces a hormone called melatonin. Does anyone know what melatonin does?
Does it have something to do with sleep?
Yes. Yeah, you've maybe heard of melatonin or seen it even. Melatonin is sold over the counter as a sleep aid. Melatonin is a hormone that is naturally released as you go to sleep. It is produced in larger quantities at nighttime when it's dark. Also in the epithalamus, we see a production of CSF, is cerebrospinal fluid. So that is produced in the epithalamus. Okay. The thalamus, kind of in the middle section, is a relay point for sensory information and some motor information. And then the hypothalamus is something that we will talk about quite a bit actually in the hormone section, in the endocrine system. The hypothalamus does a whole bunch of stuff. It regulates a lot of subconscious and unconscious and involuntary actions. It also helps release a lot of hormones. Busy hypothalamus. It also can produce emotion and behavioral drives. Super interesting. And the neatest thing about the hypothalamus is we don't know when it's working or what it does its own thing. And that's pretty much what we see for all of these midsection of the brain here is that whole area. Oh, that's my alarm. Gosh, I'm so glad I set my alarm because I did not think we were anywhere close to at that time. Okay, let me stop recording here.